Hello, our team makes microplastics identification and quantification system, and we'll be walking you through our microfluidic device, which counts and sorts microplastics. So where does the problem of microplastics come from? So there are about 8 million tons of plastic wastes which are deposited into the oceans annually. These plastics break down into smaller particles of less than 5 millimeters in size, and then they're then classified as microplastics. Once these are in the ocean, you can imagine they can be ingested by aquatic life, and then any other organism which consumes fish, which is aquatic life, which is part of the food chain, may be affected. So there are a number of studies going on how microplastics, might affect organisms. And so we found that the best way to actually combat this problem is to assist with microplastic research efforts so that as more data is backed up and analyzed, the government actually has more solid data to reference as they make better and more effective policies to combat the problem. So in the research process, one of the main things researchers do is count microplastics and also identify them. So while they're counting them, it's typically like a manual process where people have to maybe make educated guesses, estimate, or they have to individually count particles. So additionally, when they're identifying them, they have to use maybe spectroscopy, which may not always be accessible to many laboratories. Some existing solutions are gravimetric analysis, which is from the picture, you can see that it's like they would take a weight of a sample and then compare it to maybe the standard weight of one microplastic, an average weight, and then find like what the number of microplastics in their sample could be. The now red staining visual analysis method involves using a red dye. This one is particularly useful in that it will distinguish between the microplastics and organic material, but you would actually still have to visually, like individually count like which microplastics have been dyed and then record them. With gas chromatography, the microplastics are heated up, and then once they're in gaseous form, they're illuminated. Then that light is used for viewing the spectra and then identifying and counting the microplastics. With spectroscopy, it's using infrared for spectroscopy, and the FTIR machine, we're going to mention it later on as well, which is quite an expensive tool, but it's also accurate and it's used by labs with access to it. So we talked to a number of stakeholders to really gain more insights on this problem. And one of them is a professor who is researching in the influx of microplastics in Kerguelen. And her data collection took her 2 to 3 days, and she reported that she had to count each dyed microplastic piece individually when she was now post-processing the samples. Similarly, one of the environmental toxicologists from Delaware River Basin Commission that we talked to said that while he was doing his research, post-processing, he had to individually pick plastics from seas and then analyze them in the FTIR machine. So now we look at the stakeholders that we're interviewing. Most of these were, university researchers from Penn, Cornell, and we also had government workers as well who were working on this problem.
So the microplastics research process that we gleaned from the stakeholders that we spoke to can be broken up into 4 main steps. Microplastics are first extracted from water bodies by using Newston nets, or any kind of net that can grab particles from the water normally falling across the bottom of the water. This can also be done using deep water pumps, where a researcher can pump water from any part of the water column that they're interested in. And some studies will also just simply bottle water from wherever in the water they are. Simply scoop up some water out of wherever they're sampling. After a sample is extracted from the body of water, it is processed by being cleaned using oxidative agents. And normally this cleaning process is done to remove organic material to make it easier to count microplastics in the sample later on. And that step is the enumeration step. This enumeration is normally done through visual analysis, normally under a normal optical microscope, and it is sometimes aided by more expensive equipment like a scanning electron microscope, but that is normally a second step in the enumeration process, where a bulk sample is cleared out of anything that's not microplastics, and then whatever is considered microplastics is moved into a smaller glass slide or something like that and put under an SEM so that a researcher can look at it. And this step is very lengthy, very time consuming, because it is normally done all by hand. Plastics are individually picked out of the sample that is enumerated and prepared for chemical identification. This is another lengthy, time consuming step where plastics are individually analyzed using spectrometry of different forms, whether it be FTIR or Raman spectrometry, or it's gas chromatography paired with mass spectrometry. And this step is used to identify the chemical composition of the plastics that are collected. Looking at this entire process, we've decided that the insertion point of the MIQS system should be on the enumeration step. Initially, the team had planned to take over the two most time-consuming steps, the enumeration and chemical identification, and handle those with the MIQS system. However, after rescoping and looking at the size of the problem space, we redecided that the MIQS could probably best do its work if it focused only on the enumeration phase. And what we're planning to do is replace the enumeration phase with a device that can automatically count the particle samples that have already been cleaned through the processing step beforehand. And it will sort the particles that are found here into size groups, which would make it easier for researchers to do chemical identification, as they will already have particles sorted by whether or not they're microplastics and also by size. So our stakeholders expressed several main needs for a device that could do as the things that we mentioned earlier, this automatic counting of microplastics in a sample. The main things that they asked for were a product that could provide a standard for microplastic enumeration, as there's currently none or few standards that are actually written. They wanted something that could quickly produce a sample, a microplastic sample that is ready for chemical analysis. The project should also be inexpensive, as it is difficult for researchers to get their hands on things that are currently made to do this automatic counting, this automatic enumeration of microplastics in a sample. And they should also capture a range of microplastic sizes that we will define in a minute. And as an added desire, a few of the researchers that we spoke to were already familiar with a project that Penn students had done in the past that was called the Oculus. And it also dealt with standardizing microplastic research steps. So they wanted these researchers desired something that would take over and follow up with this Oculus project well. So looking at the quantified characteristics for the MIQS device that we gathered from stakeholder conversations, we have 5 main characteristics that we would like to meet. First is that the MIQS should analyze particles that range from 5 millimeters in size down to about 100 microns in size. It should be able to process a sample within 8 hours. It should be 80% accurate at identifying a microplastic in a group of organic or sediment materials. It should cost less than $5,000, and it should require less than an hour of human interaction time with the device to get one's results. Looking at several 2x2s can help to understand the current solutions that exist and the niches that they exist in. First, looking at the accuracy of these devices versus the automation that they introduce into the system, we can start on the bottom left quadrant, where things are inaccurate and also completely manual. The things that have these characteristics are microscopic visual analysis and gravimetric analysis, where both are completely manual, completely done by hand, and neither can provide a completely accurate understanding of the difference between plastics and other particles like sediment and organic materials. The gas chromatography, Raman microscopy, and FTIR microscopy are all very accurate, completely the gold standard of determining whether something is a plastic or any other kind of material. However, because a person must put in each plastic sample into the machines individually, it is completely manual. The MIQS system hopes to fit into a space that is accurately providing results to researchers while being completely automated to allow them to work on other research while their samples process. We compare the throughput of the device to the cost that it incurs. And here, again, FTIR, gas chromatography, and Raman microscopy systems all sit in the upper left corner as being low throughput and also very expensive, and at sometimes out of reach of a lab, depending on the budget that they have. And again, on the bottom left quadrant, there is low throughput and also low price, where microscopic visual analysis seems to fit that. It is low throughput as it requires a researcher to individually analyze each plastic piece in a sample that will have many plastic pieces in it, from tens to hundreds to thousands, depending on where they are collecting from. And it is still inexpensive, as most labs that this team has spoken to have access to at least an optical microscope. And finally, on the right corner, the MIQS system shares the high throughput and inexpensive quadrant with the gravimetric analysis, where anything that helps a researcher to do gravimetric analysis does tend to have high throughput, as it allows a researcher to analyze a bulk sample for microplastics and get a general mass of all microplastics in the sample. And it is inexpensive, as the materials that are required to do gravimetric analysis are normally found in any kind of chemistry lab. And the MIQS system hopes to share this space with gravimetric analysis and be similarly high throughput and inexpensive for researchers' use.
So, as Tony said, there are 5 parameters that our stakeholders wanted us to reach. Reaching microplastic sizes of 100 micrometers to 5 millimeters, costing less than $5,000, having less than 1 hour interaction time, 8-hour time to process a sample, and distinguishing microplastics with 80% efficiency. We considered 4 high-level solutions, which were gravimetric analysis, null-red microscopy, computer vision, and microfluidics. And out of these 4, only 2 of them, computer vision and microfluidics, were able to reach that. However, because initially we were considering doing both identification and counting, we put computer vision on hold because microfluidics enabled us to do a larger range of things. With the microfluidics down selection, what we wanted to do was to consider various ways of using physics to solve the problem and consider various forms of microfluidics separation that allows us to do this. One of the options that we considered was one known as acousphoresis, and this process was removed, was not considered, was not taken in because it requires a lot of large systems that are needed to basically read the reflectance of sound wave of microplastics to determine their size and their density. Magnetophoresis is another way that we could have gone, but because plastics are not easily magnetized, it also presents a harder solution for us to take. Dielectrophoresis is another particle sorting and typing method that we could use, but this one requires that you have custom electrode patterns for each design, which makes analysis much more complex. Also, because the dielectrophoresis force depends on both the type and the size of the particle, it's hard for you to distinguish these things if you don't have a uniform solution of particles that either have the same type to distinguish their sizes or the same size to distinguish their types. Capacitance-based sensing has a similar problem to dielectrophoresis in that you have 3 coupled variables that mean that you would have to have particles that either have 2 of the same of one variable so that you can distinguish on their 3rd variable. And with inductance-based sorting, the problem was that it's hard for you to induce a current within the plastic, so you aren't able to use it for microplastics as easily. Radio frequency or reflectance counting is a similar process to acousphoresis counting, and as such has the same problems in that it can't count particles. It's harder to count particles in a cheap manner because you require large systems to be able to read the reflectances easily. Going through all of these, the 2 that we came down with was to use sedimentation microfluidics as well as a process known as resistive pulse sensing. With sedimentation microfluidics able to determine the size, sort plastics, microplastics based on their size and their density, as well as with resistive pulse sensing being able to count and size particles. So we present our final idea concept sketch. So the idea is that you'd have a system that has an inlet reservoir that goes to a pump, then to the microfluidic channel, which is connected to via a USB to a computer for data processing. Then you have an outlet reservoir which receives any waste fluid or particles after we are done working with them. When it comes to human interaction, how a researcher would interact with that process would basically be after they clean the microplastics sample and evaporate the water through of their sample, outside of our system, what they would do is basically open the lid and put their microplastics in the chamber, fill it with saline solution, then plug in the input and output reservoirs before they turn on their device for sampling and for I guess working on with it. Yes, once you start doing this, the researcher ideally shouldn't interact with this process until the system has finished providing results on the counting of the microplastics. Going into the microfluidic chip, there are two systems that we would want to consider. The first system that we want to consider is the sedimentation sorting channels. This sedimentation sorting provides us a way of dealing with one of the researchers' main problem, which was that once you have, even after cleaning your samples, there is sometimes a possibility that you may miss some kind of sediments or plant material, which prevents you from being accurate with the microplastic identification. So to deal with this, we use a method known as sedimentation sorting, which basically uses the sedimentation force on a particle to sort it by its type and its density. Basically, the sedimentation force drives the particles into a streamline as they flow through the channel, and we basically drive these channels into a widening stream, which allows the streamlines to separate from each other and collect the particles based on their size and their density. As you can see, it's able to, because common plastics, plant matter, and most sediments have varying ranges of densities, it's much easier for you to use sedimentation sorting to sort microplastics out of the other kinds of plant matter and sediments that tend to be within the solution. The other part of our system is the counting. This counting process would be done using the resistive pulse sensing method, which relies on the drop in the resistance within a channel as a particle is introduced within that. So in this system, you technically have like an electrolyte solution in between two electrodes, allowing it to be freely conducting. And once a non-conducting particle is introduced there, the resistance within the electrode would increase because there is a new particle there. Yes, this system allows us to determine particles up to whose volume are up to 0.006% of the volume between the two electrodes, and allows us to determine their size as well as count them as the resistance drops occur. This presents a graph for the minimum resistance estimate, showing us how we can use the equation we showed in the previous slides to model our channel sizes and our channel design. In the first case, you have a 6 centimeter length channel that has a 6 millimeter diameter. This graph, as you can see, allows us to determine particles up to a limit of 1,600 micrometers, which is like 1.6 millimeters. But then one target range that our researchers want is that they want us to sometimes detect particles above 2.9 millimeters. In the second graph, we kind of changed the channel sizes and dimensions to see how the particle changes would occur. And here we see that the largest particle we can read is basically 500 micrometers. So you basically have a 0.6%, 0.6. So 60% drop in the increase in the resistance or change in the resistance. And here the minimum particle detection would be 168 micrometers. So from this, we can see that if we reduce our channel dimensions, we can basically reduce the minimum particles that we are sensing. So we can do all this all the way to 100 micrometers, which is the minimum that our stakeholders want us to have. So I'll now hand it over to Nyasha to talk about the circuit designs.
Thank you, Victor. Now I'll talk about the circuit design. Okay, so since we talked about the resistance changes, now we have a circuit to actually detect the resistance changes that occurs as a result of a particle passing through the electrodes. So we want to pay attention to node voltage Vx in the schematic, which is basically the part of our voltage division circuit where we'll be measuring and amplifying and then back calculating using the microprocessor. So one thing to note is that the voltage change that we're measuring that happens at the electrodes is actually going to be large enough, like at least in the range of 0.4 to 2.5 volts, such that the fact that we're using large resistors with high tolerances is not going to be a factor in us not being able to distinguish between the sizes, because again, the changes in resistances correspond to the sizes of the microplastics, which is something that will be reported to our stakeholders, to our researchers, users. So, and the way we make this voltage change large is by controlling the length of our channel. So going back to the equations that Victor was talking about, there is change increasing the length of the channel actually increases the resistance, excuse me. Okay, another important thing that I will talk about is the microcontroller down selection. And one thing here is that operating frequency was the biggest factor. We wanted the device that would provide us with the highest operating frequency possible. And so the Raspberry Pi offered the highest with 1 gigahertz. I'm going to leave it to Toni to talk about fabrication.
To ensure that the system works as intended, we plan on executing the following tests. The idea is to test each subsystem individually before putting it together and testing as a system. So starting with the pre-sorting subsystem, which makes up the sedimentation, the idea is to introduce microplastics of different sizes and different densities into the subsystem, all to fall into 4 categories: small size with high density, small size with low density, large size with high density, and large size with low density. So having 20 microplastic chip samples and having 5 samples represent each of the 4 categories. The idea is to then test how accurately the microfluidic chip is able to separate these according to both the size and the density. Looking at the pumping system, the idea is to ensure that we're able to give a constant flow rate over a certain period of time, and also to ensure that we are not rupturing the microfluidic chip system. So the idea with checking the pump would be putting in a known volume of water and then letting it run for 3 hours at a set pump rate. Initially, that will be 2 milliliters per hour. And then we're going to check this flow rate for the different sizes of microfluidic chips that we will design. And then looking at the counting subsystem, the idea is to show that we'd be able to generate resistance and would be able to read the resistance that occurs due to a microplastic passing through the channel. So initially we're going to just pass saline through the channel and measure the resistance that occurs when we do that. And then we're going to introduce a microplastic into the channel and then try to read the nominal resistance for that as well. We're going to start with a 1 centimeter wide millifluidic channel, introducing a 5 millimeter microplastic flake. But as we repeat the experiment, we'll do this for different sizes of the millifluidic channels as well as different sizes of the microplastics to ensure that we're able to measure microplastics and count them within the range that the stakeholders want. And then looking at our fall semester deliverables, we've had to de-scope and look at the idea of de-scoping the project. So now we're looking at computer vision alongside or instead of the microfluidics approach. So the next steps for us is to continue having conversations with our stakeholders, particularly surveying and comparing the microfluidics and the vision system and their desirability for that. And once we have that feedback, we'd be able to make a decision on whether to focus solely on microfluidics as we've done, or if we need to change and look into computer vision. And then the steps for microfluidics, if we choose that route, would be then to develop an analytical model of the fluid motion and then validate the counting subsystem. Whereas for the computer vision, if we end up going that route, we would have to look at applicable vision algorithms and then also define the computer vision system. These are the project schedule for spring, looking at the microfluidic system. And then in conclusion, our team has realized that there are inefficiencies in and a lack of standards in microplastic research, which inherently inhibits researchers when they're trying to do their research on microplastics because of the long times it takes for them to quantify microplastics. And then we see how this is a hindrance for policymaking as well. So then our idea is to come up with a system that will count and sort microplastics. Currently with the microfluidic from the microfluidic perspective, it looks like the device that we've shown that has a pump that will pump through a microfluidic chip and will have an outflow reservoir. And looking at the fall deliverables, the main thing we would need to do is, based on the de-scoping, look at down selecting between the microfluidic and the computer vision systems. Thank you so much for listening to our presentation. We would like to give a big thank you to advisors, especially Dr. Paula Aratia, who's our faculty advisor, as well as Graham, Dr. Turner, Dr. Heimbau, Peter Szczesniak, and Peter Bruno, who are our technical advisors as well. Thank you so much.